Hello everyone, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we're going to be talking about a little bit of organic chemistry. Now, don't let that freak you out. It's just enough organic chemistry so that we can understand the biology in future videos. And so in this video, we're really going to be focused on macromolecules. Now, organic chemistry, if you ever decide to take it, if you ever need to, is really the study of carbon-based compounds. And they have some very unique properties. And so I think probably the best thing to do is let's go back and review the structure of carbon before we get into the rest of the video. So a couple videos ago, we talked about carbon, and I used this exact picture. By the way, if you haven't seen that video, I'm going to put that in the upper right-hand corner. You really should watch that before you watch this. But we said at the time, carbon in its ground state has 6 protons, 6 neutrons, 6 electrons, and it can form 4 bonds. And we learned that individual electrons in the valence or most outer shell can form bonds. And so that means carbon has some very unique properties. Now, so this is carbon, and really what organic chemists do is they study carbon-based compounds. And for us human beings, it's a good thing because, again, most of us are made of, well, not most of us, we are all made of 99% of hydrogen, oxygen, nitrogen, and this stuff here, carbon. Okay, now that we know what carbon looks like, let's dive a little bit deeper. All right, so before we get into the macromolecules, we have to understand that there are certain what are known as functional groups. And these functional groups are special compounds that can be associated with carbon. And when they define really the behavior of the compound, its chemical properties, and we give those compounds certain names based on the functional groups that are associated with it. And so the first one I want to talk about in the upper right-hand corner is a carbonyl group, and you can see that that is a carbon that is double bound to oxygen. Then on the left and the right-hand side, we can see here, like in the example of acetone, we have carbons bound on either side. But what you're looking for with a ketone is a carbon with a double bound oxygen, and usually we can see like the generic one in the upper left-hand corner, we've got these designations R, that just means that there's special groups hanging off. Usually those are going to be carbon-based. The next one is an aldehyde. We can see that it is very similar to the ketone we've already talked about. The special exception here is that we have a carbon double bound to oxygen still, but that aldehyde has a hydrogen bound in one of those positions. It's not 2 R groups. Next functional group we have here is called a methyl group, and that is when a carbon has 3 hydrogens bound to one another. And I'm actually going to show you as we get into the macromolecules. Hopefully you'll be able to pick these things out pretty easily, but right now we're just identifying them. This is a methyl group, carbon with 3 hydrogens. This one here is a disulfide bond. You see these sometimes in an amino acid or in certain organic chemicals that can be sometimes reactive, but this is a disulfide. That's 2 sulfurs bound to one another. Now this molecule is ATP. You might have heard of it. It's the energy currency inside of a cell, but I show you this because, holy cow, when you really look at this, you're going to see a number of functional groups. So, for instance, on the left-hand side, there's 3 phosphate groups. That's a phosphorus, it's got the oxygens, and the other one, OH, which we haven't talked about, it's also at the bottom center, that is a hydroxyl group. And you're actually going to see yet another group that we haven't talked about, the very top right-hand side, that nitrogen with the hydrogens, that's called an amino group. And so this one thing right here, easy to distinguish. We've got the phosphates, phosphorus with the oxygens surrounding it. You've got a hydroxyl. We actually talked about that, if you remember, when we were talking about pH in the previous video. Those are on the bottom side. And then on the upper right-hand corner, we have an amino group. And this is a really good example of one of the building blocks of one of the macromolecules we're going to talk about today. This is called an amino acid. You've probably heard of amino acids before. They make proteins. Well, why do we call them amino acids? On the left-hand side, there's your amino group. There's a central carbon. And on the right-hand side, that's a carboxyl group, otherwise known as a carboxylic acid. Every once in a while, you'll see that drawn. The one in the green on the right-hand side, you'll have that carbon, double bound to oxygen. And then that lower oxygen will sometimes have a negative after it, no hydrogen. But that one on the right-hand side, that is a carboxyl group. You're also going to see at the bottom we have this thing called an R side chain. Whenever you see R associated with a structure, usually that means that that particular position in that molecule can change, or in that compound it can change. Okay, so now that we've identified those functional groups, we're going to see how we can use those to identify macromolecules. Macro means large, large molecules. We're talking about proteins, lipids, carbohydrates, and nucleic acids. And so these things, these macromolecules, can form polymers. The exception being lipids, they don't, but our proteins, carbs, and our nucleic acids, they can form long chains. And so how do they form and how do they break down? Well, this picture does a pretty good job of illustrating that. A dehydration reaction is one in which we form larger macromolecules. You can add monomers. Mono means single. So you can add a monomer to a polymer and you can form this long chain of macromolecules. So dehydration reaction, what you need to know is it's used to form macromolecules. Hydrolysis, lysis means to split, hydro means water, and so we're using water. Usually some enzymes are involved in either one of these reactions, but in hydrolysis what we're doing is we're splitting macromolecules into monomers. And so let me give you a really good example of why this matters to us in real life. When you consume food, the digestion actually starts in your mouth, goes into your stomach, you get more digestion, into your small intestine, even more. Well, all of that are forms of hydrolysis, breaking down the foods into small monomers, amino acids, individual carbohydrates like glucose, individual nucleotides, and then the lipids. Once those individual compounds get into your body, the cells need to reform things like proteins, complex carbohydrates, nucleic acids, and they do that via dehydration. All right, so we're going to talk about proteins first. And one of the things that we need to understand about proteins is they are vital and they form many, many different roles inside of our cells. They can be enzymes. They are involved in communication, cell recognition, structure. They can be used for energy. They can be used for transport. And so proteins are incredibly important inside of a cell. So why don't we go ahead and start with proteins, the first of the macromolecules. And proteins, as I've already mentioned, are made of amino acids. And we have the amino acid table here. There's 20 of them. I generally do not expect my students to memorize all 20 amino acids and their structures, but if you ever take a biochemistry class, you may very well have to do that. What I do expect, and what I hope many of you will get out of this, is that amino acids have certain properties depending on that R group. So if you remember, let's go back real fast. I want to show you that generic picture of an amino acid. And so if you remember, we had the amino group, the central carbon, the carboxyl group, and that upper hydrogen. That's always the same. That doesn't change. But what does change, what separates the amino acids from one another, the reason why we have 20 different amino acids, is that R side chain. So now that we know that amino acid structure, look at this picture here. We've got an amino group on the left-hand side, carboxylic acid on the right-hand side. We have our central carbon, but now what's changed is that R designation, we actually have a chemical structure there. And again, it's that chemical structure that provides the amino acid, gives the amino acid its properties. Now, what we have to understand here is, well, these amino acids are going to act differently in an aqueous solution depending on that R group. And so we see here all of the amino acids in orange. They all have something in common. If you look closely at the R group, you're going to see that the R group consists of what are known as hydrocarbons, just carbons and hydrogens pretty much exclusively. And because of that, hydrogen and carbons bound together, they're going to form non-polar covalent bonds. Now we said in an earlier video that in an aqueous solution, non-polar covalent bonds, one, are very strong, but because they're sharing the electrons equally, there's absolutely no charge, and therefore they're going to be non-polar or hydrophobic. In other words, they are not going to interact with water at all. So one of the things we have to understand is whenever you have molecules that don't have a charge at all, they're going to be hydrophobic, which means water-fearing, and that also means that they are going to separate from water. They're going to be water-fearing. They don't want anything to do with water. That's all of the hydrocarbons that we see in the orange. Now let's take a look at the yellow. We're going to notice that the yellow has something different going on, and that is, well, in most cases we have an oxygen present. And we said earlier in an earlier video that oxygen is very electronegative and it likes to pull electrons closer to itself. And therefore, there's a dipole in that R group, just like there is in water. Well, what that means is that oxygen is going to take on a slightly negative charge, and so it's going to be attracted to the slight positive charges of the hydrogen within water molecules. And so what we need to know is in that those amino acids in the yellow box, those are all examples of R groups that are hydrophilic, water-loving. Now, for the bottom row in the green and the blue, What's different about them is they actually take on a full charge. Nearly impossible to see here because these pictures are so small, but trust me, in the green and the blue, there's either going to be a full charge, full negative, or full positive. And those at the bottom are highly hydrophilic, water-loving, because that opposite charge is going to be very, very strongly attracted to water. So if the R group has a positive charge, it's going to be attracted to the negatively charged, partially negatively charged oxygen in the water. If the R group has a negative, it's going to be attracted to the slight positive charge of the hydrogens of water. And so when it comes to whether or not an amino acid is hydrophobic or hydrophilic, it's all about the R group. The ones in the orange, all hydrophobic because they are hydrocarbons, nonpolar covalent bonds, no charge. The yellow, we have an oxygen. There's a dipole there because of a polar covalent bond. There's going to be, all of those are going to be hydrophilic. The greens and the blues, because there's a full-on charge, are going to be highly hydrophilic and very strongly attracted to water. Why does this matter? It matters because when an amino acid sequence is put together to form a protein, the hydrophobic amino acids, as you could imagine, they're all going to want to be in the center. I'm looking for a piece of paper. I want you to imagine that this is a sequence of amino acids. It's a protein that is about to be made, and proteins need to maintain a three-dimensional form. So what we have is this protein is eventually going to fold up on itself in a three-dimensional structure. I'm just going to crumple it up into a ball. And now we have this protein. It's a three-dimensional structure. The hydrophobic amino acids are all going to be in the center, and the hydrophilic ones are going to be around the perimeter because that's what's going to be exposed either to the aqueous solution outside the cell or the aqueous solution inside the cell. If the amino acids are in the wrong position, and we have hydrophobic ones out here or hydrophilic ones in the center, this thing is going to start to fall apart. And so it's really important that the sequence of amino acids is proper so that the protein folds up in its proper three-dimensional shape. And so proteins do have a number of different structures. The first one is the primary structure, and that is just a sequence of amino acids. And then based on that sequence and based on the R groups, proteins will often fold up kind of on their own into either an alpha helix, or a beta pleated sheet. And so these are types of secondary structures, alpha helix or beta pleated sheet. And then, sometimes with a little bit of help, a protein will fold up in a tertiary structure, and that's the three-dimensional structure I just showed you. And that three-dimensional structure is critical to the protein's function. Not always, but sometimes, multiple three-dimensional tertiary structures will bind together to form a quaternary structure. So that's the general protein structure. Now, when proteins are in a temperature that is not ideal or a pH that's not ideal, they can denature. And what that means is a protein loses its three-dimensional structure. And as I've already said, when a protein loses its three-dimensional structure, it loses its function. So that's not a good thing. So you can see here we've got a normal protein, we get denaturation, it falls apart. Could there be a renaturation process? Maybe, but usually when it's due to heat or due to pH range that is not ideal for the protein, that denaturation is going to be relatively permanent. And so now we understand the basics of proteins. Let's switch gears, talk about carbohydrates, also extremely important. As you know, they're utilized for energy. They can be structural components, especially for organisms that have a cell wall, and they can also be used for cell-to-cell recognition. Now, carbohydrates are often divided based on the number of monomers that are connected and the number of carbons within that carbohydrate. But just like proteins, we have potentially a macromolecule that is carbon-based. It is an organic compound. And I am not going to go over every single one of these different carbohydrates, but it's just here to show you that we can see carbohydrates, the very top row. We have things like glucose, which is a 6-carbon carbohydrate. Then we have fructose, which is a different structure. Point is, you can have carbs with different structures and a different number of monomers connected. And so you can have monosaccharides, which is 1 carbohydrate, disaccharides like sucrose and lactose, which are 2. You can have polysaccharides, which are things like starch and cellulose. And some of these things we can break down easily, some we can't. So how would you know that you're even looking at a carbohydrate if these things weren't labeled? Look at the number of hydroxyl groups all over this structure. And it's very unlike the other macromolecules we're talking about. Remember, the proteins had amino acids, amino group, central carbon, carboxylic acid. Carbohydrates, there are OHs, hydroxyl groups all over the place. The next macromolecule, well, that's the nucleic acid, makes up DNA and RNA. Let's take a closer look. All right, so this picture shows us both DNA and RNA. DNA stands for deoxyribonucleic acid, RNA, ribonucleic acid. And so when it comes to nucleotides, they have three main parts, and it's the way that we identify them. We can see that we have a phosphate group, we have a sugar, that's in the light blue, and then we have in the upper right-hand corner a nitrogenous base. And so all nucleotides have those three things: phosphate, sugar, base. And again, that's why it's so important to be able to recognize the functional groups, that we recognize, oh, there's a phosphate group. We can see here we've got the sugar. Now, there are differences between DNA and RNA, and one of the differences is, again, DNA is deoxyribonucleic acid, RNA, ribonucleic acid. And if you look on the left-hand side, you're going to notice the structural difference between the two is the sugar. The sugar on the bottom has one additional oxygen compared to the sugar at the top. And so that's why it's called ribo. Ribose is the sugar, ribonucleic acid. Deoxy means that an oxygen has been removed from the sugar in DNA. There are some other important differences between DNA and RNA as well. DNA is basically the structure that holds all of the information inside of our cells. In a eukaryotic organism, you find that inside the nucleus. In a prokaryotic organism, it's in the cytoplasm, but DNA is double-stranded. It is a double helix. You can see that structure there on the left-hand side. And it has four different nitrogenous bases. It has adenine, thymine, guanine, and cytosine. So we say A's, T's, C's, and G's. Those are the four nucleotides that are associated with DNA. RNA, well, RNA is a little bit different. RNA is usually single-stranded, and it also has four nucleotides: adenine, uracil, guanine, and cytosine. So we say for the RNA, it has A's, U's, C's, and G's. And so the bases will be different between the two. And the RNA generally holds information, at least in a eukaryotic cell, it holds information for one gene that will make, generally speaking, one protein. RNA is also single-stranded, and it tends to be a lot shorter because it's not holding all the genetic information inside the cell. Now, something else that you should notice from this picture, back to DNA, it is double-stranded. And you're going to notice on the far left-hand side that when you have a G in one strand of DNA, a cross from it will always be a C. And so G always pairs with C and A's with T's. So you can see that all along the left-hand side. And the reason for that is our A's and our G's are what are known as purines. And purines are double-ringed nitrogenous bases. Our C's and T's are pyrimidines. They're single-ringed structures, and so what happens is you always have a double-ring structure pairing with a single-ring structure. The other thing that you're going to notice on the lower right-hand side is that A's and T's, there's going to be two bonds between them, and the G's and C's will actually have three bonds between them. Anytime there is a mismatch, you're going to get a discontinuity or a change in the shape of the DNA, and that's going to cause problems. And we have abilities to proofread that and to fix those genetic mutations. Last one up is going to be the lipids of our macromolecules. It's the only one that doesn't form polymers, but they're still really, really important. They can be used for energy, insulation, communication. They can also be, they are really the structure that forms our cell membranes. So let's take a closer look. And so here we have the three major types of lipids, the triglycerides, and those are primarily used for energy. We have the phospholipids, and I'm going to show you another picture of this in a second, and those are what makes up cell membranes. And then we have the cholesterol over here on the right-hand side. And you're going to see that looks a lot different. The cholesterol is involved in communication, and we also have things like estrogen and testosterone that would fit into that as well. Now, one of the things that we should notice is anytime you see a structure like we do in cholesterol, where we have a hexagon or a pentagon, and we just see lines, organic chemists assume that we know that at the points of where two lines come together, there would be a carbon there. And if you see two lines, there's a double bond. They also assume that anytime you have a point there where we should recognize that there is a carbon, and we know that carbon can form four bonds, that there would be hydrogens sticking off of there if necessary. And so what we notice with the lipids is the lipids are packed full of hydrocarbons. That's a term I use when describing the R groups of amino acids, but a hydrocarbon is simply carbons and hydrogens bound together. Not to be confused with carbohydrates, and students confuse these two all the time. Carbohydrates, lots of hydroxyl groups everywhere. Hydrocarbons, we don't really see that. And if we do, it's very, very sparingly. But hydrocarbons are what makes up lipids. And because, again, we have all of these non-polar covalent bonds between the carbons and hydrogens, they are not going to interact with water. They are, in fact, hydrophobic. And I think a lot of us know that oil and fat will separate from water. Well, this is why. Because there's nothing in this molecule, at least in the triglyceride or in the cholesterol, that's really going to interact with water. So the phospholipid is a little bit different. That top region, that is hydrophilic because of that phosphate group and that nitrogen. And then the bottom region has those hydrocarbons. Those are hydrophilic. And we're going to see how that plays into membrane structure in a few minutes. Now, another thing to make note of is we go back over here to the left. We've got the fatty acids, saturated and unsaturated. You may have heard those terms used to describe diets in the past. Saturated fats are saturated with hydrogens. So you can see single bonds, carbons and hydrogens all the way down. Unsaturated fats, they aren't saturated with hydrogens, and that's because some of the carbons have a double bond, and therefore there's no hydrogen sticking off of that other side, and they will usually be liquid at room temperature, where the saturated fats will often be solids at room temperature. Saturated fats on the left-hand side are usually animal fats. The unsaturated are usually from vegetables. And so I wanted to show you this because this is a really good example of phospholipids in a cell membrane, and we can see the structure here. We've got those little yellow hydrophilic heads that are either facing the aqueous environment or the cytoplasm, which also has water in it, and so that charge is working out really, really well. And the hydrocarbon tails, which are hydrophobic, are all facing the center and facing one another away from water. This provides the cell membrane with some very unique properties, something that we're going to talk about in a future video. All right, everyone, so that was a brief overview of macromolecules. We're going to be talking about these molecules in much greater detail in future videos. As always, thank you so much for being here. Have a great week.